Welcome back. This is your last video to review for the Chapter 17 notes. So last video we talked about translation, but we just talked about this first stage, the initiation stage, and we discussed the differences between prokaryotes and eukaryotes. But in general, initiation for both of them includes mRNA first bonding to the small subunit of a ribosome. Remember that small subunit doesn't catalyze any reactions. Its whole job is just to bind to mRNA. Once that start codon and once that methionine or modified methionine attaches, then that's when the large subunit kind of latches on and gets in place. So the next step, now that we've initiated it, is elongation. You want to make this protein huge. We started with methionine. Well, now what? So in elongation, it's actually similar between prokaryotes and eukaryotes. So the first step, well, you need to kind of match up these codons and anticodons. You know, mRNA has the code. So mRNA says GAA. Now tRNAs are attracted to the ribosome, but the ones that can make really, close to perfect fits, remember, it doesn't have to be 100% perfect because of that wobble hypothesis, but those that can make a really good fit, they're going to come in to that A site, the amino acyl site, or the amino acyl tRNA, a tRNA that has a brand new amino acid on it. So it comes in, and that anticodon that we find on the tRNA is going to form hydrogen bonds with this codon on the mRNA. Same type of hydrogen bonds we've talked about all semester long, but specifically when talking about DNA synthesis, mRNA synthesis, and now creating these proteins. Now this does require energy, and in this case we usually use the form GTP. And remember, GTP, just like ATP, it's one of those modified nucleotides that have extra phosphate groups. So now we have something coming in to that A site. The next step is, now that you have a new amino acid, you want to form a peptide bond. So a peptide bond is formed between this A site amino acid and this growing chain that is really being held in place at the P site. And you can see that here. Here's this peptide bond forming between the amino acid here in the A site and the amino acid here in the P site. In this case, we're still talking about the beginning of our creation of proteins, so it's just FMET. This should also tell you that we're looking at bacteria right now because there's that FMET. If this was a eukaryote, it would just be regular MET or regular methionine. Now the creation of this peptide bond is done by the RNA that's in this ribosome. So again, really interesting because we've learned so much about proteins working as enzymes, working as catalyzing reactions. But in actuality, when it comes to protein synthesis, it's actually RNA that is making these proteins, that's forming these peptide bonds. So even more support for the RNA world hypothesis that everything we have actually originated from RNA. So now we've hooked together these two amino acids. The next step is that the ribosome is actually going to move. We're kind of, pulling that mRNA along because, well, we need to free up that active site. We need to free up that amino acyl site. So that mRNA kind of gets tugged, and you can see this here. The GAA was here in the A site. Well, now it's being moved to the P site. We're pulling that mRNA. So we've moved a codon ahead, or 3 nucleotides ahead, and so you can kind of start it all over again. Now you're going to get a new tRNA coming into that A spot. That new tRNA has an amino acid that's going to form a peptide bond with the P group. And then once that peptide bond's formed, you're going to pull on that mRNA that frees open the A spot. Just keep doing that over and over and over again. And what you can see is with this mRNA, we're moving it from the 5' to the 3' end. And that's what we've just been talking about all class and talking about synthesis. It's always 5' to 3', 5' to 3'. You know, mRNA was copied 5' to 3', so it makes sense that the ribosome is going to read it 5' to 3'. 5' will have the start codon, 3' will have the stop codon. So now we've gone through, we've elongated for however long it is. As a note, in prokaryotes can add about 20 amino acids a second. So very fast protein synthesis. Whereas eukaryotes, we're a little bit slower. We're only adding about 2 amino acids a second. Still fairly quickly, but that is kind of a minor difference between the elongation state and both of our types of cells. Now the last step is termination, because this protein doesn't go on forever. At some point it does have to end. So looking at this first image, here's the E site. Remember, this is a used up tRNA, so it's going to get out of there. This P site is holding on to this polypeptide chain. And now we just hit a stop codon. We hit a code that there is no tRNA. It's just kind of a blank. Now, although we don't have a tRNA, or at least a tRNA holding an amino acid, something else comes and binds. It's called a release factor. It doesn't have an amino acid, but it's a molecule that can come in. It fits onto one of those stop codons. And what it does is it's actually going to catalyze this reaction, the reaction of cleaving off this last amino acid off of its tRNA. Because this mRNA is not moving anymore. So there's no cleaving except by this release factor. So the release factor comes in, it bonds to that stop codon, and it's going to cut off the last amino acid, which is going to free that polypeptide chain. That's going to free the protein. The protein can now leave the ribosome. It can start folding, it can go into the cell, it can go to the EI, it can go all sorts of places. Now that this chain has left, the tRNA that was in the exit site, the tRNA that was in the P site, they can leave too. You've gotten rid of really all the parts of translation, so the very last step is just this ribosome kind of not necessarily dissolving. It will be reused, but the small subunit detaches, the large subunit detaches, and then you can start it all over again. Now we have a couple more slides, but I do want to note there is a video down here at the bottom that overviews translation. And translation is very similar in eukaryotes and prokaryotes. There are not any differences that we're going to talk about. Now, one difference, not necessarily specifically in any step in translation, but as I touched on a couple times, is that in prokaryotes, you don't have a separation of translation and transcription. Prokaryotes don't have any organelles, so they're all kind of in the same area. So what's really cool that bacteria can do that we can't do, that eukaryotes can't do, they can actually do transcription and translation at the same exact time. So looking at this micrograph, this thin line in the middle, that's DNA. Then what you see, these little strands coming off, that's representing mRNA. That's mRNA being copied. Now remember, mRNA doesn't have to be processed. That mRNA is ready to go. So you actually have these black dots, which are multiple ribosomes. Many different ribosomes are copying that mRNA as it's being made. So bacteria can make tons and tons of proteins really, really quickly because as mRNA is being made, it's being translated by ribosomes, and it's not just one ribosome, it's many, many ribosomes. This is just a more animated version. Again, here is that DNA at the bottom. Here's RNA polymerase that's going to make mRNA. As this mRNA is being made, you have this protein coming out of it. So transcription is happening at the same time as translation, both of which happening in the cytoplasm of a bacterial cell. Now in eukaryotes, as we've mentioned before, they're separated. Transcription happens in the nucleus, translation happens in the cytoplasm. So you can't have that coupling that we saw with bacteria. However, there's still kind of this multifaceted process where it's not just one RNA polymerase. It's not just one ribosome. So the picture you see here, there's a thin strand down the middle. That's the mRNA. All of these little dots are ribosomes. Those multiple hundreds of ribosomes, translating that same molecule of mRNA. So one started and it started moving, then another ribosome attached and started moving, and another one attached and it started moving. And so you're getting tons and tons of proteins out of one molecule of mRNA. So it's a really good way to make a lot of proteins at the same time. And again, you see that down here too. So looking at this image, you can identify where the start codon is. The start codon, it says 5', but see how small those proteins are? But as you get down in this mRNA, as you get further along that code, these protein chains are getting longer and longer because you've already coded for tons and tons of amino acids already. So this is the last slide. This is an overview specifically of eukaryotes. The way I know it's eukaryotes, you see a nucleus, you see exons and introns, you see this cap and this poly A. I think that might be all the info on this one. Yeah, but do know there's other differences too. So thinking about that regular methionine versus modified methionine. The major point, not only should you know the major steps involved in translation and transcription, but also make sure you know the difference between prokaryotes and eukaryotes. They're very similar. There's nothing that's largely different between the two of them. But it is important to know kind of those small things. I could give you a picture and say, which one is this? How do you know? Or I could say, describe translation in prokaryotes. And then I would only want you to tell me about prokaryotes. So with that, that's the end. Be sure to fill out that sheet. Really, it's just a helpful tool for you. Go back and watch that video that I pointed out a couple slides ago, that overview translation. If you guys have any questions, you can ask me after the lab or lecture exam or send me an email. Good luck with everything and let me know if you need any help.